Hi folks. So today we're going to talk about the light reactions of photosynthesis. Before we get into that, let's quickly review what we learned last time, which is that the purpose of photosynthesis is to generate sugars, specifically a sugar known as G3P. And while some of that G3P, some of that carbohydrate is used for cellular respiration, ideally as a plant, you are doing far more photosynthesis than cellular respiration, because if you are doing so, then you have enough stuff to build more plant. So this allows for growth. If you're doing more photosynthesis than cellular respiration, you can grow. And then the follow-up to that is remembering that, well, to grow, you can't just grow with sugar. There's a lot more to a plant than sugar. Well, that's a lot of it. There's a lot more to a plant than sugar. So while some of this will allow us to build more sugars, more starches, and more fiber, we're also going to build the other components of living things, meaning amino acids, phospholipids, nucleotides, and more that are not even listed here. And for those molecules, which include not just carbon, hydrogen, and oxygen, we're going to need to pull up various sources of nitrogen and phosphates from the soil solution. Okay, so how do we build the sugar in the first place? There are two sets of reactions that go into photosynthesis. The Calvin cycle is the second set of reactions, and that's what actually builds the sugar from CO2. The take home from last time with regard to the Calvin cycle is to remember that we are doing a synthesis reaction. We're building something, which means that this is to some extent endergonic. And because we are doing the opposite of cellular respiration, we are adding electrons to our carbohydrate as it's going through this process. It is a reduction reaction. So the big picture for today is that we need to generate ATP to drive the endergonic reaction and NADPH to provide the electrons for this process. So those are our two things that we're going to need to create in the light reactions. So now, having erased the board, what we're going to do is we're going to zoom into a small location of a chloroplast, namely right here, and we're going to look at the membrane where the light reactions happen. The Calvin cycle happens in the stroma, which you can think of as kind of the cytosol of this chloroplast. But the light reactions are going to take place on these interior membranes known as the thylakoids. So if we zoom in, that is, into that location, let us look up close at a thylakoid membrane. And so this thylakoid membrane is going to be highly folded. Remember, this is going to be out here, the stroma. This is where the Calvin cycle is going to take place, in kind of almost like the cytosol of a chloroplast. So zooming in here, remember we have to generate two things, ATP, NADPH. I'll list them over here so we don't forget them. The ATP is going to be generated exactly as we have learned before. Nothing special. The ATP is going to be generated via ATP synthases, which I'm going to draw as their characteristic lollipop shape, right, found here. So they will generate our ATP, and hopefully you recall that what drives the ATP synthase is a proton gradient. So we're going to need to generate a proton gradient from here to here, from the thylakoid space to the stroma. Right? Same as a mitochondria, same as various mitochondria. Now, how do we generate that proton gradient? Same thing as before. Hooray. This is going to be the exact same process, slightly different proteins, but we're still going to use an electron transport chain system. The components are a little bit different. They do have different names, but conceptually, we've got the same thing going. So we're going to have a series of proteins. I'm going to draw primarily one large protein here. And these are going to be our electron transport components that are going to pump our protons into the thylakoid space. So I'll just draw that in. So protons are getting pumped into this space. So protons this way, protons this way. Well, we're going to need some electrons to fuel this, and the electrons are going to be excited from our new complexes for today. Our new complexes are known as photosystems, and these are definitely the most important new protein complexes for today. And they are large and complex. So this is a photosystem right here. And this one is known, and there's going to be another one over here, which we'll talk about in a moment. And because biology hates you, they have numbered these photosystems in a hilarious manner. And that this is the first photosystem that we're going to use, and it is known as Photosystem, 2. So this is Photosystem 2. That is the first photosystem. This over here is the second photosystem and is known as Photosystem 1. Why, you might ask? Well, this was discovered first and therefore was given and then this one was later discovered. Therefore, this was numbered number 2. And then later on, research discovered that this is the first one used in this pathway. So what are photosystems? Well, the purpose of a photosystem is primarily to absorb light. So what we're going to do is the photosystem is where we have the light absorbing pigments. Hopefully you know some of these words. Maybe you've heard of chlorophyll before. So what's going to happen is light energy is going to be absorbed by the photosystems. Light energy is going to be absorbed by chlorophyll molecules that are dispersed within this giant protein complex. That energy is going to be absorbed, causing electrons to basically jump in a quantized manner. That doesn't make a whole lot of sense. Just think of the electrons being excited by light energy. Those electrons are going to get passed, and I'll just draw them as getting excited. I'll draw an arrow like that. And then those electrons are going to get passed from photosystem 2 to this mobile carrier, which sends them to the next electron transport chain component, which is the actual proton pump in this region, to the next mobile carrier, which takes those electrons to photosystem 1, where they are going to get excited again. We'll talk about that in a moment. All right, so let me recap again. In the thylakoid membranes of a mitochondrion, not in the matrix. The matrix is where we have the Calvin cycle. So this is the matrix out here. Well, I just called it the matrix. Sorry, that is the mitochondrial term. This is the stroma. Out here in the stroma is where we get the Calvin cycle occurring, where the sugars are actually built. These membranes here are the thylakoid membranes. This is where we're doing the light reaction. And the light reaction begins with the light being absorbed by the chlorophylls within these photosystem protein complexes. The light accepts electrons, excites electrons, which are effectively excited off of the chlorophyll molecules and passed from component to component to component to component, leading to the generation of a proton gradient. Protons are highly concentrated in these thylakoid spaces. They are going to pass through the ATP synthase in this direction, and this is going to generate our ATP. Okay, well that unfortunately is the easy part of this, if you're going to label anything as easy. So because this has a lot of review, we're generating ATP from ADP, we're using a proton gradient to drive that. The proton gradient is generated by ETCs. Okay, what's new here? Well, what's new is the photosystems that are generating, that are accepting the light energy and exciting electrons along the way. But there are two things that we haven't talked about. This is the review stuff for the most part, with a couple new things. The first question is, where do these electrons come from? And lastly, where do they go? Don't forget, we do have to produce this. So the question for the light reactions, where are electrons coming from and where are they going? Now, the way I've described it so far is light excites electrons off of the chlorophylls. So the chlorophylls lose their electrons, and they move along, etc. This wouldn't remain chlorophyll. If you lose your electrons, you don't work anymore. So we can't just keep on exciting electrons off of chlorophyll, effectively making chlorophyll not itself anymore. What we do need to do is constantly be providing electrons back to chlorophyll. So this is where photosystem 2 is actually the second coolest protein complex in the known universe. I think the ATP synthase is definitely number one. Photosystem 2 is a close runner-up. Photosystem 2 is a giant protein complex, and it actually absorbs light in two manners. One reason that excites light is to excite electrons. The other reason it absorbs light energy is to, and this is going to sound ridiculous, rip water apart. I'll state that again. Photosystem 2 has two reactions that it undergoes, two sets of reactions that it is responsible for. The first reaction that it does is known as photolysis. It actually takes water and destroys it, rips it into its substituent parts. For example, water is taken, it is ripped apart, the electrons then pass and replace any electrons lost by chlorophylls. So the electrons are the ones that are really going to get excited. These electrons are coming from water. The water doesn't exist anymore. The other thing you should know is, well, if we've ripped the electrons off, there are probably going to be some protons left over, and that's true. When we rip the electrons away from water, that does deposit some protons here, and that just makes this proton gradient even better. Hooray. We keep on adding more protons to this space in this manner. And then the last thing you should realize is, wait, what about that oxygen? Oh yeah, that's the oxygen, folks. That's the actual oxygen that is released by plants. The oxygen that is released by plants is oxygen that is generated from the destruction of water molecules, as insane as that sounds. So water is ripped apart into its substituent parts. The waste product is the O2. Yes, I am fully aware this doesn't balance out. That's just fine for today. So to overview, light reactions. Light is absorbed by photosystem 2. All of these reactions are happening on the thylakoid membranes, the folds of the inner membranes of the chloroplast. The light at photosystem 2 is used for two reactions. The first reaction is to rip water apart and extract its electrons. Those electrons, which go to chlorophyll, are then excited by another photon of light energy coming from solar radiation. These electrons, now excited, are passed down an electron transport chain to generate a proton gradient, which drives the ATP synthase. Last bit, what's this photosystem 1 thing about? Well, there are actually multiple reasons, but for the purposes of today, the photosystem 1 is going to take those electrons, excite them one more time, and then pass them off to a subsidiary enzyme called ferredoxin NADP reductase. You don't need to know the specifics of that. All you need to know is that this subsidiary enzyme is going to take NADP plus and give it the electrons that just got passed along, generating NADPH. Here are our two products that we need to drive the Calvin cycle. Light plus water rips apart that water into oxygen, and then the electrons are used to generate a gradient that generates ATP and to reduce NADP into NADPH. That's it for today, folks. Feel free to rewind and rewatch as necessary. I will see you next time.